So I would like to thank the organization committee to give me this opportunity to present the last few, I would say, results that we have obtained in my group related to the drug nanocarriers. And of course, we apply it in medicine. So saying that, the first part would be related to the basic for the nanomaterials, and that would be followed with some application related to drug delivery. So first slide is to give you perspective of the different type of what we call nanomaterials related to nanoparticles. So we can see from 1963 it was discovered nanodiamond, and that was being followed by the fullerene, and after that the multi-wall carbon nanotube, single-wall nanotube, etc., up to the nanodot. And you can see that when we think about fullerene, this is zero dimension. Same thing for nanodots. And for the single wall, the multi-wall nanotube, this is related to second dimension. Now, the third dimension is related mainly mesoporous materials. So the point I want to make is about the background on the carbon dots. It was discovered in 2004 when there was, I would say, between two electrodes, the carbon ash, and they were able to, based on the electrophoresis data, that to see two bright spots, and the expected was related to nanotube, but in fact it was related to what we call in 2006 carbon dots. Part A is on the right side. We can see that when you shine light, white light, onto the carbon dots, so we can see using a filter at 450, 500, 550, etc., different emission of light. And same type of experiment with part B, but using monochromatic light to shine onto the carbon dot solution. And we can see the same behavior in terms of at 400, 450, 500, etc., the luminescence. So this work was based on Sun and co-worker at Clemson University. So that was the beginning of really description of the carbon dots. How can we do the preparation of these carbon dots? So there are two approaches, the top-down approach or the bottom-up approach. And the top-down approach is related with three different methodology: laser ablation, electrochemical, and arc discharge. That means we have to start with large molecule or large system like fullerene or ash, I would say carbon, and to reduce it to a small particle that we call carbon dot. The other approach is the bottom-up approach, using small molecule, one which is used quite extensively, citric acid, plus an amine, for example, a small molecule. And if we use ultrasonic, hydrothermal, or microwave pyrolysis, there is a possibility to prepare the small nanoparticle, but lower, I would say, in terms of size, less than 10 nanometer. So that's the different approach for the synthesis. Why it is so important now to study the carbon dots? Well, because of the size, which are very small, 1 to 10 nanometer, the photoluminescence property are very high, with high biocompatibility for different, I would say, biological system, and the functionality on the surface of carbon dots, they are great, many functional groups. And in terms of preparation, again, I mean, it takes a few days to prepare it, but maybe longer to purify the carbon dots. And we have quite important electron donor and acceptor as lead get onto the carbon dots. So if we go now for this way to look about the structure of the carbon dots, what concerns me at least is to see what is really the core, the central part of the carbon dots. And in our group, we have done the preparation of three types of carbon dots: LeBlanc carbon dots, the carbon nitride dots, and the yellow carbon dots. And we find out using Raman spectroscopy, X-ray diffraction, also with FTIR spectroscopy, X-ray photoelectron spectroscopy, so different type of methodology, and we find out that the core for the black carbon dots is mainly sp2 carbon atoms. And for the carbon nitride, we find out that there is introduction of the nitrogen and oxygen into the core lattice. And finally, very surprising about the yellow carbon dots, because we could see polymerization in 2D and 3D. And the crystallinity is very, very important. Just keep you informed that the black carbon dust was the what I call top-down approach using pyrolysis, and carbon nitride was the bottom-up approach using microwave, and finally yellow carbon dust by sonication. Again, this is the bottom-up approach. Depending on the methodology, we can see that the core of the carbon dust is really different. We cannot say that we have a uniform model for different type of carbon dust, but it has to be specific depending on the way to prepare it. So the next slide is given to show that we have a possibility to cross the blood-brain barrier using carbon dust, and the model we used was mainly zebrafish. And based on the morphology of zebrafish, we knew that when we looked the observation area, which is related to the central canal, if there is or not a passage to the blood-brain barrier. And that's important for different diseases, as you know, like Alzheimer's disease, for example, Richardson disease, Parkinson disease, and so on. Next slide will show you that when we use the carbon dust prepared from glucose, and we are able to conjugate fluorescein. Why? Because we want to see if our nanoparticle cross the BBB, and on the top the left side, the top image, the central canal is shown with the passage of our nanoparticles into the BBB. And this is very, very important, because nanoparticles shows the passage or the presence of the nanoparticle into the central canal. That means we are already crossed the BBB. So the next slide, it will be shown about another application about the carbon nitride dots that we have conjugated the doxorubicin, and mainly to look about the chemotherapy for lymphoma. So what is very important for all the carbon dust that we prepare, and with, of course, conjugation with any drug, first of all, is we have our carbon dust on the right side, and carbon dust with the doxorubicin having the NH2 functionality, so can interact conjugated by covalent bonding using the ADC NHS amidation. And as you know, as you can see on the right side, top of the slide, that we have a possibility to conjugate also the transferrin, which is a protein with NH2 group, and of course with amidation, again, process with the carbon dust. We have the system, doxorubicin, and transferrin. Why transferrin is so important? Because transferrin recognizes the transferrin receptor from cancer cells. So this is what we use on the next slide to have a look on the effect of this system onto the different cell lines. As you can see, we have 5 different cell lines, and we can see also for the doxorubicin, carbon nitride doxorubicin. The next one is carbon nitride with TF, and carbon nitride doxorubicin and transferrin. And the only one that really gave a huge, I would say, cytotoxicity to eliminate the cancer cells, this is the carbon nitride with doxorubicin and transferrin, as we can see for each of the cancer lines that we have shown with the blue line. So this is for the in vitro experiment. About the in vivo experiments using mice and using this system, on the next slide, using this system we inject the system into the vein of the vein of the mice. And on the graph at the bottom, we can see this green. This is a tumor volume against days, and the green line is when we have the carbon nitride with doxorubicin, and the red one is when we use just the system we mentioned with carbon nitride, doxorubicin, and transferrin. And we can see there is a huge reduction of the tumor volume. That means that was very efficient when we inject our system nanocarrier to the tail of the mice. So that's an efficacy to reduce the lymphoma in the body of the mice. So next slide in terms of application was a mitochondria. Why? Because as you know, mitochondria in the cell structure is in the cytoplasm, and as shown for this mitochondria, that this is the energy, I would say, that dissipate this energy into the different site or different organelle needed to function. So what we used was the metformin with citric acid, which is the bottom-up approach, and having this reaction using microwave at 7 minutes, for 7 minutes at 1000 watt. So we can produce our carbon dust, and with this, I would say, centrifugation and filtration, and followed by dialysis, we can separate our carbon dust, and we can use a dry, I would say, methodology to obtain the metformin carbon dust. So on the next slide, this is the characterization of these carbon dust. And this is true when we think about characterization. Whatever the nanoparticles we prepare, we are using the same methodology. That means absorption, fluorescence, and Fourier transform infrared spectroscopy. For the absorption, as you can see, we have the 340 nanometer band, which is due to N-pi transition, 275 also N-pi transition, and finally the N-pi at and also pi at 225 nanometer. Saying that, we can see also for the fluorescence, looking at the normalization at the bottom on the right side of this slide, that this is excitation wavelength dependent. So the exciting from 360 to 540, so we have a shift of the emission from roughly 450 to around 650 nanometer. And finally, we use the Fourier transform infrared spectroscopy to know about what is the functionality on the surface. And there's a correlation between FTIR and also X-ray photoelectron spectroscopy to identify really the functionality on the ligand, as a ligand on the carbon dust. The size of the particles, we have done this determination by AFM, which has a high resolution for Z-axis, and also the transmission electron microscopy, which has a very good resolution on XY axis. So we can see that in terms of characterization on the next slide, that on this side we can see that the average of the size is around 3 nanometer, having both methodology. So we can apply it with the cells structure, and just in the middle, when we think about or look about the red channel for mitochondria staining. So the control, we don't see much about the mitochondria, but 1 hour treatment with 5 millimolar of met carbon dots, so we can see using also DAPI, the mitochondria very well demonstrated. And same thing also when we leave the cells overnight with 2.5 millimolar, so we reduce the concentration of metformin carbon dust with the Mitro tracker, the TAPI. So we can see this is specifically staining mitochondria. So the next slide shows this, the reason why we have this, I would say, carbon dust that goes into the cells is related to the fact that the met carbon dust is electropositively charged, and we know that the inside of the wall of the mitochondria is negatively charged. So here is this interaction, positive negative interaction, that's favor to bring the metformin carbon dust into the mitochondria. So that's the hypothesis that we have to explain this very high concentration of the met carbon dust into the mitochondria. So the next one is related to another type of carbon dust. This is a yellow carbon dust. And we know that this yellow carbon dust can inhibit high beta secretion, APP also interaction. So the way to prepare it is again, bottom-up approach using citric acid and 1,2-phenylenediamine, and we use a solvent water. And sonegation is related to with a frequency of 42 kilohertz for 1 hour. And again, for the characterization of the particles, as I mentioned previously, absorption, fluorescence, X-ray photoelectron spectroscopy that will link to ATR, FTR. And in this particular case, you can see this is no more excitation dependent, but excitation independent. Going back to the UV rays absorption, so the band, this is what will correspond, will know about the absorption for the C double bond O and also the pi-pi interaction. Good correlation between FTRR and XPS measurements in terms of the presence of ligands onto the carbon dust. And finally, the atomic force microscopy give us a very good, I would say, very good size of a 3 nanometer for the Z, I would say, axis. And on the next slide is it was promising to observe that amphiphilic yellow carbon dust is able to cross the blood-brain barrier. And on the slide, by the bottom of the slide, part B and C, we can see this inhibition using these yellow carbon dust of the APP and also the amyloids, and the treatments between, I would say, without any carbon dust and a 10 micromolar is quite important, in particular with the APP. So the next slide is to mention that the conclusion of this work is related to the fact that glucose-based carbon dust can cross the BBB using zebrafish as an in vivo model. Second, what is important to, I would say, have toxicity to eliminate the cancer cell is to use carbon nitride with doxorubicin and transferrin. Transferrin is used because we have, for many cancer cells, overexpression of the transferrin. That's the reason why this is so efficient. Third part is related to metformin, which is a new preparation, and this metformin carbon dust really targets specifically mitochondria. So there is a possibility to apply it as a drug nanocarrier specifically for mitochondria. And finally, the yellow carbon dust has the possibility to cross the BBB. And we know that there is a passive diffusion and inhibits the production of APP and also A-beta. Important because knowing that yellow carbon dust can cross the BBB, we can use it eventually as a nanocarrier against Alzheimer's disease using the, I would say, amyloids hypothesis. So what is important for the yellow? This is promising also for an antiviral agent. So that's very important. So just to mention that those who pay the bill, I think it's very important with NIH, NSF, and University of Miami, and Abdulaziz also University. And we have many collaboration as I listed, and my group without any graduate students, undergraduate students to work in our group, it will be difficult to give the result that I presented today. So, again, I thank you for your attention, and I hope that I will be able once upon a time to see you face to face in the next conference. Thank you very much.